In this video, we will discuss about structure of monosporic embryo sac. In the previous videos, we have discussed how embryo sac is produced, what are different types of embryo sacs, and what is megasporogenesis. Now, in this video, we will study about structure of monosporic embryo sac. Now, if we see angiospermic flower, angiosperms are the plants which bear the flowers. In them, this part is the gynoecium, right? And this is differentiated into ovary. This is style and this is the stigma. Now, this ovary part has ovules. Inside the ovule is present the embryo sac. This whole structure is also called as carpel. So this is the ovary which is having ovules inside it, and inside the ovules are present embryo sac. Now if we see the structure of the ovule, now this is the ovule which is having, this part is called as nucellus, and these are the coverings called as integuments. This is inner integument, this is outer integument. And this part is made up of mass of parenchymatous tissue called as nucellus. Now this is called as nucellus. Now inside this nucellus is present this mature embryo sac. Now why this is called as monosporic embryo sac? Because in here, at this point in young condition, a cell is differentiated. This is called as archisporial cell. This we have discussed in the previous video in detail. That archisporial cell then differentiate into megaspore mother cell. Now that megaspore mother cell, this is megaspore mother cell. Now this megaspore mother cell nucleus is diploid. Now this will undergo meiotic cell divisions to form 4 haploid nuclei. Now then wall formation will take place and we'll get 4 megaspores. Now out of the 4, 3 they degenerate. Now this is toward the micropillar end of the ovule. This is toward the chalazal end of the ovule. So toward the micropillar end, 3 megaspore, this and this will degenerate, and one which is toward the chalazal and this will enlarge and become functional. So this is the functional megaspore. So out of the 4 megaspore, 3 degenerates, 1 become functional. So we call it as monosporic. I mean, out of the 4, only 1 spore is participating in the formation of the embryo sac. So this is called as monosporic type of embryo sac. Now this will develop into mature embryo sac. What is the structure of monosporic embryo sac? We'll discuss here. Now this is the mature embryo sac present in the ovule, like we discussed here. This is the embryo sac which is present here in the nucellus, and this part of the ovule is micropillar end, this part is the chalazal end. Now we can see here what is the structure of monosporic embryo sac. Now this is the embryo sac. Now this has at this end toward the micropillar end, this has egg apparatus. Egg apparatus is basically made up of egg and synergids. And we can see here these are finger-like projections. These are called as filiform apparatus. Now this part is having 2 polar nuclei, and here we have 3 antipodal cells toward chalazal end. And this is the large vacuole present in the embryo sac called as central vacuole. So we can say it has single egg or oosphere, then it has 2 synergids, then 3 antipodal cell, and single large central cell having 2 polar nuclei. So basically its arrangement is 3 plus 2 plus 3. 3 forming egg apparatus, 2 forming polar nuclei, and 3 forming antipodal cells. Now if we see the structure of mature embryo sac or the gametophyte, we can see here. This is the structure of the mature embryo sac. We can see again this part is egg, right? This is also called as female gametophyte because this functional megaspore has been produced after meiosis. So all the nuclei will be haploid in nature. So this is the egg cell which is having nucleus, and this is the vacuole present toward the micropillar end. And this is being assisted by these 2 cells called as synergids. Now synergids, they have helping cells, they are called as helper cells. They have filiform apparatus which are finger-like projections present over here. So these filiform apparatus will help in guiding the pollen tube, like pollen tube when comes during fertilization. So this will guide toward the embryo sac, and this pollen tube will reach toward the egg cell with the help of these synergids. And this also acts as a shock absorber when pollen tube enters. So this will act as shock absorber, as well as this help in obtaining nourishment from the nucellus, because this is present inside the nucellus. So this will help in absorption of the nourishment for the developing female gametophyte. This will be done by the synergids. So basically synergids have filiform apparatus. This is the nucleus of the synergids, and this is the vacuole of the synergids. So these synergids, they are helping cell. So they help the egg or assist the egg in the fertilization process. Now these are two polar nuclei present in the center, and these are three antipodal cells present here at the chalazion end, and this whole cell is called as central cell, and these are the tiny vacuoles which later on can get organized to form a full-fledged bigger vacuole. So this is the structure of mature embryo sac, and I will discuss in detail one by one. So we can say embryo sac is single egg or oosphere which is present toward the micropillar end. And if we see the egg cell as well as these, now supposing this is the egg cell and these are two synergids which are present over here. Now they are interconnected with each other, right? Their wall is common, so they are interconnected with each other, and these connections, they are called as plasmodesmatal connections, by which their cytoplasmic continuation is maintained. But when this egg is fertilized, after fertilization, egg will develop its own wall. This will develop its own wall, and its nucleus will also become diploid in nature. First it is haploid. After fertilization, this will become diploid. This will discuss again in the next videos. Now these are two synergids like we discussed, which are present, and they are helping the egg cell or assisting in the fertilization. So two synergids are there. They have filiform apparatus, right? Filiform apparatus, as we discussed, this will help as a shock absorber. This will help in transportation of the nutrient toward the egg cell. This will also guide the pollen tube toward the egg cell. So this synergids, they are helping the egg in fertilization process. Then we have three antipodal cells present over here toward the chalazion end. When egg is fertilized, these antipodal cell will degenerate, right? And they will also help in absorption of the nutrient from the nucellus tissue. Like this is present inside the nucellus, so antipodal cell will help in degenerate, add nutrients, as well as they help in absorption of the nutrients from the nucellar tissue. Then we have this central cell which is single, and it is having two polar nuclei, right? Now when pollen tube comes, this carries the, like this is the pollen grain, and this will germinate to form the pollen tube, and pollen tube carries the two male gametes, right? One male gamete will fuse with the egg cell, and second will fuse with the polar nuclei. So one will fuse over here and one will fuse over here, right? This is also called as a double fertilization, because two male gametes, they are fusing one with the egg and second with the polar nuclei. Now first two polar nuclei, they have one, this is single nucleus, this single, so this is 2n, we can say. But when single male gamete again comes, and this will become triploid in nature. So this nucleus will become triploid, and when this become triploid, this will develop into endosperm. Basically, endosperm is the nutritive tissue. So this egg which is fertilized, this will develop into embryo, and polar nuclei after fertilization become triploid in nature, and they will develop into the endosperm. So this is about the functions of polar nuclei, antipodal cell, and synergids, which are helping the female gametophyte in fertilization process. So this is all about structure of monosporic embryo sac. Thank you for watching my video. If you like my video, please like, share, and subscribe. Thank you.